Good afternoon to Doctor and all of my fellow friends. For today oral presentations, I'm going to talk about algae management in biofuel production. My name is Francisca Denlawing, BS19110344. So first of all, what is algae? Algae are organisms that grows in aquatic environment and it use light and carbon dioxide to create biomass. So there are two classifications of algae, microalgae and macroalgae. Macroalgae, which are measured in inches, are the large multicellular algae that often seen growing in ponds. These larger algae can grow in a variety of ways. The largest multicellular algae are called seaweed, and examples is like giant cap plants, which can be more than 100 feet long. Second one is microalgae. One of the other hands are measured in micrometers and tiny unicellular algae that normally grows in suspensions within a body of water. Next is biofuel. Biofuel or any form of fuel that is derived from biomass of plants, algae, materials, or animal waste. Since such feedstock materials can be replenished readily, biofuels is considered to be a source of renewable energy, unlike fossil fuels such as petroleum, coal, and natural gas. Here are some examples for biofuels: wood, used directly as a raw material that is burned to produce heat, and heat turned and used to power plants to produce electricity. Ethanol, which primarily by fermenting starch or sugar. Methane gas, derived from a decomposition of biomass in the absence of oxygen. Next is the current potentials of microalgae for use as a biofuel. So this means by what potentials microalgae have able to use as in biofuels. Firstly, the algae biomass contain three main components: carbohydrate, proteins, and lipids, also known as natural oils. So natural oils from the microalgae is produced in form of triglycerols, which is the right shape of oil in producing biofuels. So different algae might have a different ways in generating new energy, and some algae species can produce hydrogen gas under specialized growth conditions, and some algae can be burned similar to wood or anaerobically digested to produce methane biogas. Besides that, some algae biomass can also be treated by pyrolysis to generate crude bio-oil. So next is the biological and adaptation. Here are some few reasons for why microalgae is used in the algae to biofuel arena. First, microalgae grow quickly and contain high oil content compared with terrestrial crops. So basically, they take a season to grow and only contain a maximum about 5% dry weight of oil, dry weight of natural oil. So as for microalgae, they commonly double in size every 24 hours. That gave a huge difference between productions of natural oil. So during the peak growth phase, some microalgae can double the productions of natural oil every 3.5 hours. Oil content of microalgae is usually between 20% and 50% weight, while some strains can reach as high as 80%. Now, now is for productions and agronomic informations. So there are two types of macroalgae in algae collections: photoautotropics and heterotrophics. But due to high capital and operational costs, heterotrophic algae cultures is hard to justify for biodiesel productions. And to minimize the cost, algae biofuel productions usually relies on photoautotropic culture that use sunlight as a free source of light. And as for phototrophic microalgae, require light, carbon dioxide, water, and inorganic salts to grow. The culture temperature should be between 15 to 30 degrees Celsius for an optimal growth. As for growth medium, it must contribute the inorganic elements that helps make the algae cells grow, such as nitrogen, phosphorus, iron, and sometimes silicon. So for large scales of microalgae productions, algae cells are continuously mixed to prevent the algae biomass from settling, and nutrients are provided during daylight hours when the algae are reproducing. However, one of the quarter of algae biomass produced during the day can be lost through respiration during the night. So here, a variety of photoautotropic-based microalgae culture systems are available. For example, the algae can be grown in a suspensions or attached on solid surface, but each system has its own advantage and disadvantages. So currently, the suspensions-based open ponds and enclosed photobioreactors are commonly used for algae biofuel productions. For suspension-based open ponds, in general, is an open pond is simply a series raceway outside, while photobioreactor is a sophisticated reactors design which can be placed indoor, either in a greenhouse or outdoors. So the details of the two systems I will describe one by one. So the first one is suspensions-based open ponds. Open ponds are the oldest and the simplest system for mass cultivations of microalgae. In this system, the shallow ponds is usually about 1 foot deep, and the algae are cultured under conditions identical to the natural environment. So the environment has to be as natural as it's come from. The pond is designed in a raceway configuration, in which paddle wheels provide circulation and mixing of algae cells and nutrients. The raceways are typically made from poured concrete, or they simply dug into the earth and lined with plastic to prevent the ground from soaking uh, up up the liquid. So baffles in the channel guide the flows around the bands in order to minimize the space. Algae broth is harvested behind paddle wheel after it has circulated through the loop. The next is the enclosed photobioreactor, which has been employed to overcome the contaminations and evaporation problem encountered in open ponds. These systems are made of transparent materials and generally placed outdoors for illuminations by natural light. So as for enclosed systems, the algae biomass is prevented from settling by maintaining a highly turbulent flow within the reactor using either mechanical pump or an air-lift pump. A portion of the algae is usually harvested after the solar collection tube. In an open raceway system, this is not a problem as oxygen is simply returned to the atmosphere. However, in a closed system, photobioreactors, the oxygen levels will build up until they inhibit the poisons, the algae. So next is the advantage and disadvantages of the two systems. The advantages is they can overcome the problems in contaminations, evaporations, and content in open ponds. That is the advantages for bioreactors. The biomass productivity of bioreactors can be 13 times greater than that of a traditional raceway ponds on average. Harvesting of biomass from bioreactors is less expensive than that from a raceway pond, since the typical algae biomass is about 30 times as concentrated as the biomass found in the raceway. However, enclosed bioreactors also have disadvantages. For examples, the reactors has more expensive and difficult to scale up. Moreover, light limitations cannot be entirely overcome since light penetration is inversely proportional to the cell's concentration. Attachment of cells to the tube walls may also prevent light penetration into the bioreactors. Although enclosed systems can be enhance the biomass concentrations, the growth of microalgae is still suboptimal due to variations of temperatures and light and intensity. So, in both systems or productions of mercury in biofuel concepts, the Department of Energy has concluded that the algae biofuel production was still too expensive to commercialize in the near future. Here are three major factors limiting commercial algae productions that exist: the difficulty of maintaining desirable species in the culture system, the low yield of algae oil, and the high cost of harvesting the algae biomass. So in recent years, algae biofuels production has gained renewed interest. So both university research group and startup businesses are researching developing new methods to improve the algae processes efficiency with a final goals of commercial algae biofuel production. The effort and development can be categorized into several areas, including the increasing oil contents of existing strains or selecting new strains with high oil content, increasing growth rate of algae, developing robust algae growing system in either an open air environment or an enclosed environment, co-product development other than the oil, using algae in bioremediations, developing an efficient oil extraction method, and one way to achieve all these goals is to genetically and metabolically alter algae species. The other is to develop new, improved existing growth technologies so that the same goals listed above are met. So besides in producing biofuel, algae can also be explored for a variety of other uses such as fertilizer and pollution control. Certain species of algae can be land for use as an organic fertilizer, either in its raw or semi-decomposed form. Algae can be grown in ponds to collect fertilizers run from farm, and the nutrient-rich algae can be collected and reapplied as fertilizer, potentially reducing the crop production cost. Moreover, in wastewater treatment facilities, microalgae can be used to reduce the amounts of chemical needed to clean and purify water. So for the summary of my presentation today, microalgae are an ideal biodiesel feedstock which eventually could replace um, fuels due to several advantages um, such as high oil content, high rates of production, and less land, and so on. Currently, our gas biodiesel production is too expensive to be commercialized, but due to the static costs associated with oil extraction and biodiesel processing, and the variability of algae biomass productions, cost-saving efforts for algae oil production should focus on the production method of the oil-rich algae itself. So here are some reference I took for my presentations today, and that's all from me.